Principles of Inheritance and Variation, Session 3. In this module, we will learn about certain terminologies which we learn in genetics. The first terminology we are going to learn is a factor or a gene, the functional unit of heredity which is responsible for expression of a character in a progeny. We know that during sexual reproduction or during any reproduction, the characters from the parents gets passed on to the offsprings, and it is the genes which helps to transmit the characters from the parents to offsprings. So the functional unit of heredity which is responsible for expression of character in the progeny is called as a factor or a gene. We know genes are located on specific structures called as chromosome. So the position of a gene in a chromosome is called as a locus. So locus is the position that a gene holds on a chromosome. Then the third term is an allele, the alternative form of a gene for a contrasting character present on identical locus of homologous chromosomes are called as alleles. A character is anything which we inherit from our parent. So alternative forms of character are called as traits. So character, if we take character as height or color, then the traits of the character height are dwarf and tall. And the traits for color of a flower are pink, white, violet. All these are the traits. So just like that, a character is coded by a gene, and the traits, the alternative forms of genes are called as alleles. Is it clear? So alternative forms of character are called as traits, and alternative forms of a gene is called as alleles. So alternative form of gene for a contrasting character present on identical locus of homologous chromosome. We know homologous chromosome we have studied in class 11. It is the position. It is the position, then the structure, the shape of the two chromosomes we inherit, one from a mother and one from a father, is called as a homologous chromosome. So a pair of chromosome which are identical in shape, in size, in all the genes it holds is called as a homologous chromosome. So alleles occupy the identical locus on both the chromosomes, that is a homologous chromosome. And alleles are represented by letters. Alleles are represented by letters, and for a gene there is a pair of alleles. So each gene has a pair of alleles. Phenotype. The external appearance of an organism due to an influence of genes and environmental factors is called as a phenotype. Phenotype, how an organism looks. Either it can be due to an influence, either due to an inheritance of genes or due to environmental factors. So how an organism appears is called as a phenotype. For example, the plant is tall. The girl is having curly hair. All these are the phenotype. Whereas the genetic constitution of an individual which is responsible for that particular phenotype is called as a genotype. Is it clear? So it's suppose we studied in class 10 that a phenotype tall is either represented as capital T or capital T, small t. And dwarf is represented as small t. So these capital letters, capital T, then capital T, then small letter, small t, all these represents the genotype which is responsible for that particular phenotype. The correct proportion of a phenotype in a population is called as the phenotypic ratio, and the proportion of the genotype in a population is called as the genotypic ratio. Now, there are two other terms, homozygous and heterozygous. I told you an individual has a pair of alleles, and if that individual is having an identical pair of alleles, identical genes in an allelic pair for a character, they are called as homozygous. That gene is said to be homozygous gene, and that individual is said to be an homozygote. For example, capital T, small t. They are homo means identical. So homozygous means that particular individual is having identical genes in an allelic pair. Whereas heterozygous, that individual, if it is having an dissimilar pair of alleles for that particular gene, that individual is said to be heterozygous. For example, capital T, small t, it's an heterozygous. Both the alleles are different. It is capital T and small t. The gene that expresses its character in a heterozygous condition is called as a dominant gene. For example, if the heterozygous condition is capital T and small t, what will be the phenotype? We had learned in class 10. The particular hybrid will be tall. We will be learning all this in detail in the coming modules, but yet we have to understand what is these terminologies, or else we cannot explain this each and every time when we are doing a cross. So dominant gene means the gene which expresses in a its character in heterozygous condition. So when it is capital T and small t, what is expressed, which is a character which is expressed, it is capital T, and that particular plant becomes tall. So T stands for tall. So that gene which expresses in a heterozygous condition is said to be a dominant gene, and the other gene which is hidden or which is not getting expressed in the presence of a dominant gene is called as a recessive gene. And the progeny obtained by crossing two parents which differ in characters is called as a hybrid. So hybrid has a mixing up of both the parental characters. And we have test cross and back cross in order to understand whether a progeny which is obtained is either homozygous or heterozygous. So the cross between an F1 hybrid, F1 means first filial generation. So when the two parents are crossed and the generation which is obtained in the first generation is called as a first filial generation or F1 generation. So the cross between that F1 hybrid with any one of its parent is called as a back cross. And test cross means the cross between a hybrid and the homozygous recessive parent. Recessive parent, for example, small t, it is homozygous and it is recessive because it cannot be expressed. So it is expressed only when it is in the homozygous condition, that is called as a recessive parent. Whereas in the case of capital T, the small t is not expressed. It is called as a recessive gene. And the homozygous recessive parent is crossed with an F1 hybrid. Then that type of cross is called as a test cross and is used to identify the genotype of the hybrid. Thank you.